I want to thank you all, as Alan said, coming out on a Thursday evening to hear some of the things that I'll tell you about cancer. As you said, I'm a professor at Boston College. I teach advanced biology, especially related to cancer, and some general biology as well. And that's what basically what my profession is. I'm a professor. But we do a lot of research, basic research on cancer. We've developed some of the best preclinical models that we can test and vet a variety of different approaches. And then I work with a number of physicians who are anxious to adapt what we're doing. Because my view on cancer is I think we need to vet these things clearly in models that replicate exactly what goes on in the human disease. So we're sure of two things. Number one, is what we're planning to do work? And does it not harm the subject? Because if we're able to get the disease managed without harming the subject, maybe we can now adapt this for the human, and we're starting to see some successes. What I just did was I took the data from the American Cancer Society, and we just, cases per year, new cases, deaths, and deaths per day, and we go from 90 to the last 26 years. And you know, some people say, oh, do we see a lot of good news in these numbers? The good news is that the number of people dying per day, the increase for those 26 years is not as dramatic as the number of new cases. So the new cases are flooding into the system, and not only in the United States. This is happening in all countries throughout the world. And in China, China is just devastated by cancer, more so than many other countries. So the good news is that the rate of deaths, the body count is not increasing as rapidly as the new people coming into the, and they say, well, we're making headroads. And then, of course, every now and then on the news, you're going to hear major breakthroughs in cancer that we're seeing, that for the first time, the death rate is going down. And you'll see a couple of those situations where it dropped here a little bit. And then you say, oh, we're making major. But then the next year, it goes back up again. And it's this constant increase. So you have to say to yourselves, you know, what is going on here? How is it possible that our country and so many other countries are spending so much money on this disease, and yet the progress in managing the disease is, in my mind, not nearly what we think it should be. And it seems like we've become complacent, that cancer is just something we're going to have to live with, and we're going to have to suffer with, because it's part of our technological societies. And I'm saying, no, that's not necessarily the case. So we have to then look, what I have to do is I have to introduce you to what we think cancer is. Because I think what we think cancer is currently by the establishment, the pharmaceutical, the academic industries, the federal government, may not be correct. And I'm going to present evidence and let you look at some of the evidence that we have accumulated here. Okay, so the question is, provocative question. Is cancer a nuclear genetic disease, or is it a mitochondrial metabolic disease? They're very different aspects of this. Because this is the cell, and we know that these cells grow out of control. And this is the nucleus of the cell. Now, according to the current view, the DNA right here in the nucleus becomes damaged, and that's what sends these tumor cells into uncontrolled growth. The other problem, the other aspect here is these little bean-shaped organelles that are in the cell, they're called mitochondria. And they're the organelles that generate the energy for our cells. They're responsible for our respiration. So the question is cancer a genetic involving the nucleus as the primary driver of the disease, the mutations in the nucleus, or is it possible that it's these little organelles in the cytoplasm outside of the nucleus that might be the real culprits on this? And this is the focus of the first part of what I'll be talking about, just so you have an idea of what we're really talking about here. Okay, so the current dogma is cancer is a genetic disease. In this paper by Hallmark's Next Generation, a very commonly cited paper, it pretty much sets the whole playing field of what the nature of this disease is. Cancer cells carry the oncogenic and tumor suppressor mutations that define cancer as a genetic disease. Now, we call it a dogma. A dogma is an irrefutable truth. A dogma is something that's no longer questioned. A dogma is something that now is the way it is. We have dealt with all the other issues. This is the way it is, and we're going to work within this mindset which leads to an ideology. Now, I say it's a dogma because I told you I teach biology and biochemistry and genetics at Boston College, and any of the textbooks that you open up in the section on cancer, first thing, cancer is a genetic disease caused by oncogenes and tumor suppressor genes, which I'll go over. So this is where it is, and this information is not questioned. There's no other alternative. You see, you're reading the books, the kids have all memorized. So we've indoctrinated several generations of scientists and physicians through our educational process, and therefore the research and the approaches to this disease come based on the dogma. Now, here's an example that they use in the textbooks to illustrate this, the car. The dogma is platformed on the somatic mutation theory of cancer. And in my book, I go through a very detailed explanation of all this. Basically, mutations in genes inhibit or stimulate cell division and contribute to cancer. So here's a group of cells, they're growing out of control, and this is what we call cancer. It's a bunch of cells in the body, and I'll talk about how they spread and all this other stuff, metastasis. But they use this car. The car is out of control. It's going as fast as it can. It has no brakes. And this is to represent these cells. There's no longer any growth regulation. The cells are growing like crazy because they have no inhibitor genes, and the genes that stimulate growth are out of control. So now, the research industry and our attempt to understand and manage this disease is based on these hallmarks. And these are the hallmarks that are outlined by Doug Hanahan and Robert Weinberg, who's at MIT. Now here, sustaining proliferation, evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, which is the blood vessels that go into the tumor cells, and resisting cell death. Now you have to realize each one of these hallmarks is the subject of multi-billion-dollar industries, okay? We have pharmaceutical and academic hospitals and labs focusing on one or several of these aspects, the so-called hallmarks. The concept, of course, is that if we can understand the mechanisms of these hallmarks, we can then provide better therapies for managing the disease. Now, some emerging hallmarks that have come out in the last couple of years, avoiding immune destruction, tumor-promoting inflammation, genomic instability, and dysregulated cell energetics, which I'm showing you right here, and I'll be focusing on this so-called emerging hallmark, because I don't think it's the emerging hallmark, I think it is the origin, and I'll show you evidence for that. Now, to preserve the dogma, if you read these books and these papers, these scientific papers very carefully, they recognize this as an emerging hallmark, but they say dysregulated cell energetics is now an emerging hallmark that is programmed by proliferating inducing oncogenes. This concept preserves the dogma. So the dogma, everything has to do with the nuclear gene mutations, and that's the origin of the disease. So wherever you want to look at, any one of these hallmarks or any of these differences, it's got to be based on the fundamental issue of the dogma, which is the somatic mutation theory of cancer. So what that means is that your cancers, wherever it comes, breast, colon, brain, lung, starts with a mutation. And then one mutation here in a series of chromosomes. Here's a normal cell, another mutation, another, over time, something this, that, or that. You get a certain number of mutations, and bang, you get this dysmorphic malignant cell. So it goes from a normal epithelial cell to a malignant. This is called epithelial mesenchymal transition. This is an epithelial, this is a dysmorphic cancer cell, which arose as the result of an accumulation of these different kinds of mutations. The problem, of course, is that no one's really sure how many mutations it takes to get from here to here. It could be 4, it could be 20, it could be 1,000. We're now recognizing that there could be millions of mutations present in these cancer cells. Some are considered drivers, some are considered passengers, some are considered partial drivers, backseat drivers. There's all kinds of different kinds of names that we're calling these different kinds of mutations that are thought to be driving and producing the disease. So then, as the result of this dogma and mindset, we now have what we call personalized therapy, and you'll hear this advertised, at least up in Boston at the Dana-Farber, and you'll hear it from Cancer Center Treatment Centers of America. You'll hear about, we're going to use genomic testing to see whether or not your cancer can be treated or managed or something like this. And this woman is staring into the screen looking at information with extra copies or genes or whatever, and then she's going to make a decision and say, does this information help us in prognosis, or do you think we can use this information as the result of a therapy? And the major medical schools are building big departments that are breaking down, that using the genomic information. You hear it all over. This is the dogma. The cancer is a genetic disease based on somatic mutations, and the way we're going to manage this disease is we're going to better understand how these mutations work to produce the disease.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto-for-cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto-for-cancer.
Okay, the singular most important aspect of the disease is metastasis, because ultimately metastasis is what is most difficult to control and ultimately leads, the disease itself or our attempts to manage the disease leads to the death of a majority of people with cancer. So the interesting thing, and this is a diagram or a picture, the green cells are the tumor cells, and they go through this stereotypical cascade, metastatic cascade. The interesting thing about the metastatic cascade is that it's a very stereotype. Whether you have lung cancer, colon cancer, bladder cancer, breast cancer, these cancer cells go through a very stereotypic process. Local invasion, so the cells break through their normal barrier. Intravasation, which means they enter into the bloodstream. This is a very sophisticated biological process. You can't believe the number of reactions that require cells that are not supposed to be in the bloodstream to get into the bloodstream. They have immune system survival. The immune system doesn't attack them. They're immunosuppressive, and some of these new drugs you hear about, these checkpoint inhibitors and all this, they're designed to try to make them more immunoresponsive. In extravasation, so these tumor cells here in the bloodstream, they leave the bloodstream and they enter into different organs or different systems at distant places. This is called extravasation and secondary tumor formation. And it happens when your body starts to collect tumor cells. You can no longer use surgery. You can no longer use radiation. You have to then fall back on these new so-called immunotherapies and these new old toxic drugs and whatever you want to do. You're trying to cleanse your body of these metastatic cancer cells, and then you hear all the time, well, they reverted, we weren't successful, they're coming back, these cancers are very tough, they're doing this and they're doing that, and everybody seems to be okay with that. Now, one of the questions is, how can the accumulation of random somatic mutations cause a non-random metastatic cascade based on this epithelial mesenchymal transition? Nobody discusses that. It's just one of those things we don't discuss. So I put this picture up here, the dilemma that we have. How can we advance new therapies for cancer management and prevention if the 800-pound gorilla thinks cancer is a genetic disease and dogma rules? So who is the gorilla? Who's this gorilla that's telling us? It's the National Cancer Institute. Go to their website. See what they say on the National Cancer. Cancer is a genetic disease. The academic industry, the academic and the top labs in the top medical schools are locked into the dogma. And the pharmaceutical industries. The big labs, the pharmaceutical industries, the National Cancer Institute, this is the dogma. They are of the view that cancer is a nuclear genetic disease. Therefore, all of the things that we're doing, the majority of things we're doing, are related in one way to the other to the somatic mutation theory and the dogma. So, let me present evidence that challenges the somatic mutation theory of cancer. And again, when I present this, some people don't want to see it, they don't want to talk about it, they don't want to hear it. So, but anyway, I'll proceed with it anyway. Now, one of the most interesting studies that was done by McKinnell and his group, and I had a chance to speak to him, he's a professor emeritus at the University of Minnesota. And he did this study and published in a top journal, Science, 1969. And what he did is that there's a form of tumor in the frog. It's the luck tumor. It's a renal, very deadly, rapidly growing malignant tumor in the kidney of the frog. So he took the cells out of the frog kidney, Here's the cell from the tumor cell, and he removed the nucleus of the tumor cell that supposedly has the mutations that cause the disease. And then he put that nucleus into a fertilized egg and removed the natural nucleus and put the tumor nucleus into this new cytoplasm that contained normal mitochondria. And then you have this now new egg, and this egg developed into this tadpole that's shown right here. And he evaluated the tadpole. There was no abnormal cell growth. There was no dysmorphic. There was no tumors. There was nothing that you would call cancer. Yet it came from this rapidly growing malignant tumor. The interesting thing about this study is that this tadpole, for whatever reason, there was something in the nucleus of the tumor cell that prevented the tadpole from maturing into an adult frog. So there was some problem. But the findings are inconsistent with the somatic mutation theory, which says that the genes in the nucleus are responsible for the abnormal cell growth. There was another study done by Beatrice Mintz in Philadelphia at Thomas Jefferson, and Carl Amensi, and they have a malignant teratoma, which is very rapidly growing, kind of an embryoid kind of tumor. And they took the whole cell out of that and they put it into a blastocyst of a mouse, and they cloned a mouse that contained part tumor cells, part cells from the teratoma and part normal cells. And yet the mouse had no cancer, and they were able to show that these tumor cells were not forming cancer in this mouse. So their conclusion was conversion to neoplasia, which is this abnormal cell growth, does not involve structural changes in the nuclear genome. What does that mean? This is not consistent with what we're told in the somatic mutation theory. Wasn't discussed. So this was another study that was done by Jim Morgan and his group at St. Jude's Hospital in Memphis, Tennessee. They cloned mouse embryos from the nuclei of medulloblastoma. This is a kind of a brain tumor in the cerebellum. So they took the nucleus out of the brain tumor and they put it into a stem cell, and they cloned this mouse. This is an embryoid mouse, but the ectoderm, mesoderm, and endoderm were all in their normal configurations. Now, the interesting thing, although the medulloblastoma embryos aborted, none exhibited uncontrolled proliferation resembling tumorigenesis. How is it possible that if the somatic mutation theory of the cause of the disease is in the nucleus controlling the proliferation of the cells, we take that nucleus and put it into a new cytoplasm, we don't get that phenotype? These findings are inconsistent with the somatic mutation theory of cancer. Rudy Yanish at MIT and his group did another study. Rudy is a very interesting guy. He's the leader in mammalian developmental biology. He is the premier mammalian developmental biologist. He saw those other studies and he said, I don't believe those other studies until I can do it myself with my system at MIT. So he took melanoma cells and from a mouse melanoma, and he characterized the kinds of mutations that were in the nucleus of the melanoma. Then he took the nucleus out of the melanoma cells and again put it into an embryonic stem cell and cloned these mice. This is one of them here, from the nucleus of the melanoma, and he found the same mutations that were in this mouse embryo that were present in the original melanoma, but this mouse had no cancer. It wasn't producing the phenotype that we would call neoplasia. So he says here, the presence of the major genetic abnormalities in the embryo cloned from the tumor nucleus provides unequivocal genomic evidence that these mice were cloned from the tumor nucleus, yet they did not present the phenotype that would be expected based on the dogma and the somatic mutation theory. Inconsistent. This was a more recent study done by Benny at Baylor College of Medicine, Benny Kiripatu and his group. And what he did, he took very aggressive human breast cancer, metastatic breast cancer cells. And what he did is, this is an even harder technology. He removed the cytoplasm containing the abnormal mitochondria and put normal mitochondria with still the metastatic nucleus. And before he did that, he characterized all these abnormal oncogenes, the ones that are supposedly driving the disease. And in the melanoma, they had very high oncogenes. And when he took and put normal mitochondria into this breast cancer cell, the cell stopped growing and the oncogenes turned off, which was very like, what's going on here? In other words, the oncogenes are being controlled by the mitochondria in the cytoplasm, which is like completely different from the somatic mutation theory. So what I did last summer, I finally, after years, I put it in my book and then I extended it recently. I brought together all of these observations. I only gave you a snapshot because I don't have time to go through all these experiments. But they're all in this paper and they're in my book. So what I did is I said, okay, we have two fundamental explanations for the origin of cancer. we have two fundamental explanations. One is the somatic mutation theory, and the other is it's not in the nucleus, it's in the cytoplasm and the mitochondria. Let's compare and contrast the observations from all these different kinds of studies and see what the results. And so I tell people, if you're interested in knowing the argument, read the paper, make your own decision. Don't just listen to me. Look at the original papers, read them for yourselves. I know that sometimes they can be somewhat technical, but that will, you weigh and you compare and you contrast, and then the rational mind will look at the information and say, hey, I think it's this way or that way. So this summarizes what I just said in a very simple cartoon. Normal cells beget normal cells. Tumor cells beget tumor cells. We know this. Okay, what is responsible for the dysplastic cell growth and the abnormal growth of these tumor cells? Is it the nuclear mutations? Is it the mutations in the nucleus, like the dogma says? Or is it the defects in the cytoplasm, the mitochondria? Okay, it was tested. So you'd move this nucleus into a new cytoplasm with normal mitochondria, and you got normal cells, sometimes normal tissue, and sometimes a normal mouse. And if you move the normal nucleus into the tumor cytoplasm, you got either dead cells or tumor cells. You never got normal cells. This is just the opposite of what you would expect if cancer were a nuclear genetic disease. So this is very hard information for indoctrinated people to swallow. This is very hard information to swallow. All right? Again, I don't want to hear it. I don't want to talk about it. Okay, so if somatic mutations are not the origin of cancer, then where do these cells come from? How do we get cancer if mutations in the nucleus are not the cause of the disease? So Warburg had this disease pegged a long time ago. A famous German scientist from the 1920s, did his work in the 20s and 30s, 40s, and 50s. Basically, Warburg said cancer arises from damage to the cellular respiration in cells. Energy through fermentation, which is a primitive form of energy, gradually compensates for insufficient respiration. Cancer cells continue to ferment lactic acid or lactate in the presence of oxygen. This is called the Warburg effect. You see, we shouldn't be fermenting if oxygen is present. It's called the Pasteur effect. These are very complicated phenomena. If you put yeast cells in no oxygen, they throw out lactic acid. They begin to ferment. Okay? But if you bring oxygen in, they stop fermenting and they start to breathe. Respiration is breathing. Cancer cells continue to ferment even when oxygen is present. So this is an aberration. This is an abnormal thing. All cancer cells, to one degree or another, are fermenters. All right? So then the enhanced fermentation is the signature metabolic malady of all cancer cells. So when we look at cancer from the genomic atlas, the Cancer Genome Atlas, we take someone's tumor and we spend a lot of money fragmenting thousands and thousands of mutations and cataloging all of these mutations. And what we find is that every cell in the tumor has a different constellation of mutations. So the goal of the personalized medicine is essentially to try to target different kinds of pathways from these mutated cells to try to manage the disease. But every cell has so many different kinds of mutations. How are you going to build a cocktail of targets when every cell is different and they all have constellation? This is the strategy. This is called personalized medicine, which is being done today. Now, that's one. Okay, that's what I just said. Just think for a minute. Do you think that's the wise strategy? Now, if every cell in that tumor is fermenting to some degree, regardless of what the nuclear mutations are, and we know what the fuels are that these cells are using to ferment, doesn't it make sense that you might want to deprive those cells of those fuels they're using to ferment when all of them are doing the same thing to one form or another? Why are we not doing that? So when we talk about respiration, what is respiration? We breathe air. We're sitting here right now, tonight. We're breathing, right? Nobody's fermenting. Even if you're falling asleep, you're not going to be fermenting. You're still going to be breathing. So we breathe in air and we exhale, all right? We breathe in oxygen, and the waste products of fermentation are basically carbon dioxide from the foods that we ate and water. That's the waste product, all right? And we get most of our energy by breathing. We breathe. And we get very little energy by primitive fermentation processes, this glycolysis in the cytoplasm, and we get a little fermentation energy from the Krebs cycle, the TCA, tricarboxylic acid cycle. So you can see 5%, 5%. Most of our energy comes from breathing. We breathe in air. Oxygen is what's keeping us going. The cancer cell is very different. The cancer cell gets a lot of its energy from these more primitive forms. These primitive fermentation processes existed on the planet before oxygen came 2 billion years ago. We had a lot of organisms back then, but they fermented. There was no oxygen. How were they going to grow? And they grew like crazy. They proliferated and then they died. When there was a food source, they could grow like crazy. When the food was stopped, they died. Okay? So cancer cell is simply a shift away from breathing, away from respiration, to processes involving fermentation. So I went through and I showed you, this is the mitochondria inside of an electron microscope, because these organelles are very, very tiny. You need an EM, electron microscopy, to see them. Here's the organelle broken down into its various components. These little stripes that you see here, these stripes, they're called cristae. They are those little membranes, cristae, contain the proteins and the lipids that allow us to breathe, to get energy from oxygen, all right? We breathe in, and that's electron transport chain in action, right? And those little stripes. This is the mitochondria from a glioblastoma multiforme, a very deadly human brain tumor that you've heard probably a lot about with Senator Biden's son and others that have had these horrible tumors. And there's a crystallysis. You see, those little stripes are missing. We call this a ghost mitochondria. The very structures that are responsible for us getting energy from respiration are missing. This organelle is not going to be able to produce sufficient energy by respiration. So if that cell, that tumor cell, is going to live, it has to get an energy from a different source. And that source is this primitive form of energy called fermentation. Now, I want to go back and say that we don't know of any cancer cell that has completely normal numbers and function and structure of mitochondria. When you look at tissues, in culture you might see a little bit different, but when you look at solid tissues, you usually see that. Okay, so what we have to do, if we're going to redefine what the nature of cancer is, we just can't ignore all of the other information that's happened through decades of research by the top medical schools and the top labs throughout the world. We don't ignore that stuff. We just have to re-explain it. We just have to provide an alternative explanation for what we have, a new way of looking at the same information. The information is not going to change. We have mutations in the nucleus. Yes, there are mutations in the nucleus. Yes, these cells ferment, they don't respire. Yes, they have all of these things. So how do we reconfigure the origin of the disease in light of a reanalysis of the information about the disease? So we have to go back to the hallmarks of cancer, like I showed you that the dogma is telling us, and we have to be able to re-explain the dogma in light of an alternative explanation for what the disease is. And that explanation is mitochondrial metabolic disease, but we have to first deal with the paradox. The oncogenic paradox has stumbled many scientists in this field, okay? So let me just say for a minute, what is this oncogenic paradox? It was first pointed out by Albert St. Georgi, who won the Nobel Prize for his work on vitamin C. He said, there's a problem here. There's a problem, and it's the paradox. How is it possible that so many provocative agents that we know are linked to the origin of cancer could produce cancer through a common pathophysiological mechanism? Okay, and if you want to see, if any of you have read Siddhartha Mukherjee's book, The Emperor of All Maladies, it was on the New York Times bestseller list. He won the Pulitzer Prize. Ken Burns, the famous movie guy, he just produced a film on the Emperor of All Maladies. I mean, he got a lot of press, a lot of people heard about this. Look at page 285 in Mukherjee's book. He's struggling with the oncogenic paradox. On 303, he struggles with the oncogenic paradox. Look it up. So he said, how is it possible? We know carcinogens in the environment cause cancer. Radiation causes cancer. Hypoxia, inflammation, rare mutations, RAS oncogene, viruses, age. The older you get, the more likely you get cancer. Viruses, hepatitis C virus, papilloma virus, HIV virus, they all cause cancer. They all damage the respiration. They all cause problems. They all generate reactive oxygen species in the organelle called the mitochondria that's supposed to give us energy. Okay, And then people say, well, what about the what about these people who get, like Angelina Jolie gets BRCA1, she has her breast removed, her ovary and whatever, and other people do these prophylactic responses because they inherit a gene. Therefore, cancer must be a genetic disease because the gene causes it. Li-Fraumeni syndrome, ovarian and brain cancer, and a variety of other cancers caused by inherited mutation in P53, the tumor suppressor. Those mutations knock out and influence the function of this organelle. So they're really secondary causes, as is carcinogen, radiation, and hypoxia. All of them work through the mitochondria. And the ROS, reactive oxygen species, these ROS are carcinogenic and mutagenic. That means they cause mutations and they produce cancer. So the ROS come from damage. These things in the environment or inherited damage the organelle. This produces reactive oxygen species. These reactive oxygen species further damage the organelle, and they also cause mutations. So now we go back to the dogma. The dogma says cancer is a disease of somatic mutations. Where are all these somatic mutations coming from? They're coming from the ROS generated by the mitochondria. So are they the cause of the cancer, or are they the effect of the cancer? Which I should say, not the cancer itself, but the initiating cause of the disease, because then the disease progresses. Now, these little organelles signal the nucleus and says, we're not able to get enough, we can't breathe, basically, we're suffocating. So the cell has two choices. The cell can either die, it could say, well, you suffocate, you're going to die. You hold your breath long enough, you're all going to die, right? Okay, or you can turn on genes that are going to upregulate primitive pathways that existed a long time ago, millions of years, that were evolved, and you ferment. And all cancer cells then begin to turn on these oncogenes, and the oncogenes are transcription factors to upregulate these primitive energy pathways that existed on the planet before oxygen came in. So these cells are going back to a state that organisms on the planet existed in a billion years ago. So now they become free of regulation. So what was normally regulating the mitochondria is now becoming dysfunctional. Now we can begin to link the hallmarks of the disease to this linkage scenario. As the cells stop respiring, stop breathing, they start fermenting. The red line is indicative of fermentation. So your cells, the mitochondria become progressively damaged because this is an evolving, escalating situation. Now the cells enter the default state. All of these first three hallmarks, these cells flip to the way they were before oxygen, which is dysregulated cell growth. They proliferate. As long as food is there for fermentation, these cells will grow like crazy. Then because cancer looks like a wound that doesn't heal, blood vessels come in. So this is the angiogenic thing that we spend millions of dollars, billions of dollars trying to figure out this process here, driven by VEGF. And then we get why the cells, normally when a cell gets sick, it dies. As a programmed cell death, apoptosis, the cell begins to die from a programmed process. The very organelle that is the kill switch, so to speak, the very organelle that's the kill switch to get rid of a dead cell or a sick cell is broken. The switch doesn't work. These cells are now dysregulated, and they continue to proliferate as the result of this fallback, and they're lacking it. Now, where does metastasis come from? Because that's really the thing. You can talk about, a lot of what I've already spoken about happens in benign tumors as well. And yet you can't believe how we spend more money studying benign tumors than we do studying metastatic disease. How do we get metastasis? Okay, here's the scenario. Here's a normal population of cells. They get provoked by by something in the environment or a chemical, radiation, virus infection, age, whatever. They start to proliferate, but they don't spread. They form a lump or a bump, and they continue to grow, maybe very microscopically. The cells of our immune system, the macrophages that signal that there's something disturbed in the microenvironment, come in, and they come in as wound healing cells. And the wound doesn't heal because they throw growth factors out to help heal these wounds. They don't know that these are abnormal cells, so they throw growth factors out. These cells are scripted. They're evolutionary, metabolically scripted. They don't think, they don't have minds. They do what the environment tells them to do based on the situation. Now, what happens, these cells are so effective at healing wounds that if the cells they're trying to heal the wounds don't heal, they actually are very fusogenic. In some ways, they actually fuse with each other and with other cells, say, okay, I'm going to actually take care of this by fusion. Now, what happens is organelle transfer. I said these have damaged respiration, mitochondria. Gradually, as this festation continues, the mitochondria inside these red cells, these MACs, become displaced by the mitochondria from the abnormal cancer cell, these non-metastatic cells, and now you have these cells here. These are hybrid fusion cells. These macrophages they go through our body. They are the sentinels of our body. They enter and exit tissues. They're programmed to do this. If you cut yourself and bacteria get into the skin, the macrophages come in, kill the bacteria, heal the wound. Now we have dysregulated, out of control macrophages that now they enter into intravasation, extravasation. This is what they do. They're genetic. You don't have to envision new mutations to do this. And there's a number of cancers, they can't find any mutations in these things. All right? Now, the interesting thing here is these guys are glucose and glutamine driven. Okay, their behavior is glucose and glutamine driven. That's what they do. So if most cancers express the Warburg effect as a result of impaired respiration, what therapies might be effective in managing and preventing tumors? Now, you see, once we have a clear idea of what the nature of this disease is, okay, once we know, oh my God, it's not a genetic disease, despite the fact that we're spending billions of dollars in this failed effort, and the body count tells you that. Okay, now what can we do? All right, so these cells are now dependent heavily on glucose and glutamine. So what we want to do now is we want to try to see what we can do. One strategy is to try to reduce levels of fermentable fuels while elevating levels of non-fermentable fuels. One of the ways we did this initially was calorie restriction. It's a metabolic cancer intervention, involves a total dietary restriction, differs from starvation, maintains minerals and nutrients, and enhances mitochondrial biogenesis, which is really important because you can build new mitochondria by simply stopping eating. But you got to realize this is not easy. I always said people love to talk about calorie restriction on a full stomach. Believe me, I go around. You know, even I talked to the last week, I was out talking to the Calorie Restriction Society of America. You know, those guys there, you know, they don't eat anything, right? They share a grape when they have a meeting. You know, so, but any event, it's very hard to do that. Okay, so calorie restriction in mice mimics water-only therapeutic fasting in humans. What does that mean? I just drink water. You mean I can't have a snack? No snacks, just water. All right, what about no, just water. Ooh, geez, that's pretty tough. How long do I got to do that? Can I do that for an hour or two? No, you got to do it for like a week. Oh, that's all that. I'm walking the other way. So, but anyway, you're going to see some, and we've documented differences between mouse and man because the mouse, his heart rate is 600 beats a minute. You know, if our heart rate was 600, my head would blow off my body. So, so you have to realize what we see in the mouse. Oh yeah, look at that mouse. All he has to eat is 40% less, and look, he gets his tumors gone. Doesn't work that way in us. All right. So anyway, when you stop eating, blood sugar goes down. All right. So your brain needs all this sugar. If your blood goes down too far, you fall unconscious. You got to call the EMT. Gives you Chiclet or something, makes your sugar go back up. Hey, what happened? So what happens is, but you see, it goes down gradually. It doesn't go down too fast. You stop eating, blood sugar goes down because insulin goes down. Insulin goes down because blood sugar goes down. Your body says, hey, when is this guy? Then I get hungry, I got to go back and eat some stuff. I get my sugar back up again, I feel good. All right, so, but if you don't go back and get something to eat, the body says, okay, this guy's not going to get something to eat. If we don't get something to eat soon, we're going to go unconscious. So the brain has an alternative. It burns ketones. Where do you get ketones from? Fat. Okay, we're evolving, our species, we survived. We're supremely designed to not eat. Now you put an organism, the human being, in an environment. This organism, we're designed to survive for long periods of time without food because we have an incredible capacity to store energy. What happens when you take this kind of an organism and put it in an environment where all this food is glucose, as much as you can eat, wherever you want to go, everywhere on the store, the donuts, pizzas, you get fat, right? This is what happens. Now, what happens now, you can mobilize that fat if you stop eating. The fat you take it out of your fat, goes to the liver now brings these ketone bodies, and the brain begins to burn ketone bodies. This is how we function when we don't eat. So these water-soluble ketone bodies are an alternative to glucose, and they can then replace glucose. The problem is the tumor cells can't use these very well. They need a good mitochondria to burn ketones, and the tumor cells don't have that. And here's just a quick diagram. So a brain tumor cell, we work with a lot of glioblastoma, a lot of brain tumor. Glucose comes in. Under normal conditions, the glucose would be fully oxidized and you'd produce CO2 and water. But the tumor cells can't use they can't fully oxidize or breathe, so they produce a lot of waste. Lactic acid is a waste product of glucose fermentation. Creates acidity in the microenvironment, acidity. It causes inflammation and all this kind of stuff, and the tumor gets worse. So we lower glucose and we bring in ketones. They bypass. The problem is the tumor cells can't use these ketone bodies effectively. So we're essentially starving. We're metabolically marginalizing the tumor cells. We're taking away their prime fuel, which is glucose, and we're offering them up an offering of an alternative fuel that all the normal cells in our body can burn effectively because we evolved to do that, but the tumor cell can't because it has these defective respiration. So here's an example of a mouse. This is ad libitum, means this mouse was allowed to eat all the food that it wanted. It's a high carbohydrate, standard mouse chow diet. And we just restricted the amount of calories by 40% 3 days after tumor cells were implanted into the brain. And we can get anywhere from a 65 to an 85% reduction in the size of the tumor simply by restricting the amount of calories that this mouse ate. Now, we also show each one of these little squares is a mouse under a different diet. And you can see as glucose goes down, going from higher to lower, ketones go up. This is the evolutionarily conserved adaptation. Happens in the mouse, happens in the rat, happens in humans. As glucose goes down, the tumor weight goes down. Since we published this, it's been replicated in humans for brain cancer and breast cancer and colon cancer. You eat less sugar, the tumors don't grow as fast. Now, what is the mechanism by which it works? It targets 3 of the prime hallmarks of cancer. It's anti-angiogenic, it stops the blood vessels going into the tumor. It's anti-inflammatory, and it's pro-apoptotic. It kills the tumor cells by a non-provocative mechanism. It's called programmed cell death. Each one of these processes is the subject of multi-billion-dollar industries building drugs that can be anti-angiogenic, anti-inflammatory. Bevacizumab, Avastin is one of these. You know, it doesn't work. I'm going to explain that. Anti-inflammatory drugs. If you stop eating, you get the same effect, right? Nobody tells you that. Now, I'm going to go over this. This is, we built a model of GBM, a very powerful model in mouse. It's one of the best animal models, if not the best. And we showed that if we simply this is a histological section of the tumor in the brain. You put the cells in, they go from one side of the brain to the other, just like human. And you can see when we put them on the calorie restriction, you can see how demarcated the tumor is. Here it's not demarcated. This dark blue are the tumor cells. They run right through. We can slow it down. We don't cure. We don't use this term, oh, we can cure cancer with calorie restriction, or we can cure cancer with ketogenic diets. Wrong, okay. Don't ever say, people in the media will say, Seyfried says you can cure cancer with a ketogenic diet. No, that's not true, I'm telling you. You don't do that. What you can do is you can slow the system down and make the system vulnerable to cocktails which will involve hyperbaric oxygen and a variety of other drugs. So this is one way, and I'll talk about that in a little bit. So now the problem is restricted ketogenic diet. See, the problem with calorie restriction is calorie restriction. Nobody likes calorie restriction. Cancer patient comes in, and then you tell him you can't eat anymore. The guy already has depression. He has all these problems. He's freaked out by the nature of disease, and now you say go home and don't eat anything? It's not what he wants to hear. So we have to find a way to replicate the biochemical processes in the body that calorie restriction will do without doing full calorie restriction, and that's the ketogenic diet. So restricted as well. So what is this diet? Now this is another thing that gets, what happened to the, there we go. Ketogenic diets. Yeah, listen to me. They're not all the same. Okay, we've investigated several commercial and home prepared, animal, human, whatever. All these diets are not the same. So don't anybody think about, I'm going to do a ketogenic diet. What are you going to do? The types of fats? How do you know it's going to be different from an Atkins diet? So Atkins diets, thank you. Okay, Atkins diet, see, the ketogenic diets are very low in carbs, very high in fat, moderate protein. So Atkins diets have too much protein. People say, oh, I'm going to do it. They don't know, I'm blowing ketones from a urine stick. That must be I'm in ketosis. No, you're blowing ketones in your urine. That doesn't mean you're in ketosis. You have to measure it in your blood. You have to measure it carefully in your blood. So you have to be very careful. Ketogenic diets are not Atkins diets, although Atkins diets can make it appear to have ketosis. So, and also it's the type of fats. The type of fats. So this is a very sophisticated medical procedure. It's not something to be taken lightly. People have to know about this. So when you get out and hear all these people, I'm on this ketosis, that ketosis, you got to be very careful about this. We'll give you the blood meter and you can determine for yourself. Quickly, if you eat too much ketogenic diet, your tumors can grow faster. All right? So this is another thing you have to be careful about. People say, oh, if you eat all this fat, you're going to be very unhealthy. And they're right. If you eat too much fat, you get dyslipidemia, you take your blood, it looks milky. It looks milky. This is not healthy. When you're on a good ketogenic diet, beautiful amber, HDL, LDL ratios should be beautiful, low triglycerides, low blood sugar, high ketones, blood premier. It's unbelievable how healthy your blood work can be if you do this correctly. If you don't do it correctly, you put yourself at risk and you make the tumors grow faster. And this is what we showed. The tumors grow fast on unrestricted ketogenic diets. They grow slow when you restrict the diet. And that's because blood sugar doesn't go down and ketones don't go that high. But when you restrict the diet, KDR is restricted, look how high the ketones. Now we're going to begin to choke these tumor cells. We're starving them of their fermentable fuel and we're force feeding them. They can't eat it. Now, here's the thing. Can ketogenic be effective for patients? And what about ketogenic diet as a medicine? Food as medicine. This is a novel concept in medicine today, right? Most medicine is drugs, right? Pharmacy. What about food as medicine? The ketogenic diet should be respected, and you have to know about it to use it correctly. This was the first study that was done by Linda Nebling, pioneer, give her a lot of credit for this. She rescued 2 little children that had, they were considered hopeless cases at Case Western Reserve. They did not respond well to the chemo, and they did not respond to the surgery nor the radiation. And they finally was able to rescue them and say, I think I can save these kids if I put them on a ketogenic diet, lowered blood sugar, elevated ketones, and provided long-term management to both. Quality of life increased dramatically and survival increased. One child lived several more years and then passed away, and the other child was lost to follow-up. Okay? I looked at this and I said, what is going on here? The thing that gets me upset is that the first evidence clear in humans was this study in children in the pediatric oncology ward. Okay? No other study has yet replicated this in children. We're doing it in adults, and yet I have friends that are treating the children next door in the epilepsy, pediatric epilepsy with ketogenic diets. The children, thousands and thousands of kids are being treated with ketogenic diet to manage epilepsy. And in the next wing, the pediatric oncology kids are not being given a therapy that has been shown to be very effective. Why is that? Now, we published the second paper after Linda, from a person who contacted me from Italy, Giulio Zuccoli. This into a 65-year-old woman. She had progressive memory loss, chronic headaches, and nausea. She had histology. They did a debulking, and it was clear case histology for GBM. And here you can see the MRI, a multicentric GBM. You can't cut it all out. This is one of the things with these GBMs. You can't cut them out. And besides, many of the tumor cells already invade outside of what you can see, and you can't see that under these MRIs. So what we did is, right after debulking surgery, she went on a 3-day water-only fast and then transitioned into a ketogenic diet, right, restricted. And we got her blood sugars down a little bit and her ketones up a little bit, so the sugar is black, this is red, the ketones. And then her first MRI, and she had standard of care, let me tell you, but we had poor encephaly. They call that radiological resolution. They couldn't find the tumor anymore, all right? This can happen, but it happens very rarely. So they didn't know what to say. Everybody was excited. 3 months later, she does another one. It looked the same. 3 months later, another one. So people were thinking, man, how do you feel? The woman's quality of life was unbelievable. It was good. She was swimming in the Mediterranean. She was feeling very, very good about herself. Gets off the diet. 10 weeks later, the tumor returns, all right? Rather than going back on the ketogenic diet and therapeutic fasting, she decided to do Avastin, bevacizumab, right? Which was taken off the market for breast cancer because it was causing more harm than good. So they gave, and it's still used in the United States and Switzerland, and it causes the tumor cells to spread even worse. It's terrible. In my book, I blasted it. So yet we still use it, okay? And I can go into that. I can do a discussion, but I don't have time. So then I got really upset. And I said, you know, we did a lot for this woman, and then she went on the Avastin, and then she died, and I said, Jesus, what the hell's going on here? So then I started looking at what are we doing to these cancer patients? Brain cancer. What are we doing to these people? All right? You come in, you fall down, you have a headache, you have a seizure, you have something like that. Oh my God, you got a GBM or something like that. So the first thing, we have to resect immediately. So we debulk. We go right in and we take the tumor out as quickly as we can. And it also depends on the nature of the patient. Sometimes you have watchful waiting, sometimes you don't. That's the surgeon's decision. But when you debulk a tumor, you create a tremendous amount of wounding in that place. Then as soon as the patient recovers a little bit from the debulking, they go under a very powerful radiation therapy. And it's called radiotherapy, and it causes a tremendous amount of necrosis in and around where the tumor used to be. Okay? Now, oftentimes the brain swells when you give radiation from all this, so they give high-dose steroids to stop the swelling of the brain. This drives the blood sugar up. This makes blood sugars go high. And you create all this necrosis. And this releases glutamine, a lot of glutamine. You break these glutamate cycles, and you free up these molecules. You increase glutamine. I said glutamine is a powerful driver of tumor cells. And then you elevate, and then to reduce the swelling, you put steroid, and that raises sugar. And to make matters worse, all these tumor cells, the majority of them, are infected with human cytomegalovirus. Very interesting infections. It was only recently found a few years ago. They are like superchargers for using glucose and glutamine. So we have an infection of the tumor cells, and we freed up massive amounts of the two fuels that are going to drive their fermentation. We have the standard of care. I published this in Lancet. I didn't think they would accept it, okay? It's a top cancer journal. I was like shocked when they said, we'll publish your note. I said, geez, I thought I was going to be shot. So you know what the field, here it is. I published this in 2010. And I said we create the perfect storm. We create a perfect storm for the demise of these poor patients. Now, if you stick the beast with a stick, don't you think it should elicit some sort of a response? Nothing. Silence in the entire oncology community. Nobody said a word. So now, I just said we created the perfect storm in the way we treat patients who have glioblastoma. Here are the data, okay? The survivors using standard of care. The red are those individuals that got radiation alone. 278, 0 survivors. Then we added temozolomide, an alkylating agent, which was the biggest breakthrough in brain cancer research for years, and we were able to get progression-free survival a little bit better. This gap here was considered the biggest breakthrough in brain cancer research in 50 years from this alkylating agent. And I said, wow, this is, better than just radiation alone. But then a paper came out of UCSF, San Francisco, published in Top Journal by Johnson. Temozolomide increases driver mutations. What? How is it possible? What does that mean? I said to myself, I said to my students, I said, what the hell is going on here? Okay, we're giving an alkylating agent to a brain cancer patient, and they are doing better, but this drug increases driver mutations. Isn't that the dogma says if we increase driver mutations, we increase fitness and growth speed, but yet the patients are doing better. How do you explain that? So I was again provocative. I wrote in Cancer Letters, another top journal. I said, what does temozolomide do to people? All right? Fatigue, nausea, vomiting, diarrhea. These are kind of the adverse effects that you get from temozolomide. All of that is indirect calorie restriction. I said it. All right? Silence. Now, paper just came out from Stupp, the guy, this is the Stupp protocol. He seems to be able to be the person who says, this is the way you have to do it. He just came out and said, we don't think we should use steroids any longer to treat brain cancer patients. 6 years went by before the field comes out and says, now we don't want to use steroids. All right? You shouldn't use any of this.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life.
Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paperbook via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
Now I'm going to give you another case report. Influence of metabolic therapy in an adult female. This person contacted me out of the blue. She was a marathon runner. In 2009, she had gait disturbance at 56 years of age. They misdiagnosed her for a couple of years. Then she went in, they did an MRI, and she has a tumor, diffuse infiltrating brainstem glioma. Can't operate on this. It's in a part of the brainstem. You can't touch it because you'll die. So she initiated the restricted ketogenic diet with supplements, metformin, DCA, and curcumin. She refused all recommended chemo and radiotherapy from 5 major medical centers, both in France. She lives in France. In Nice, I visited her last year. Drove us around, took us to Ez. Have you ever been to Ez? It's very nice. She drove us there with the brain tumor. So now, unfortunately, she's starting to, this October, she'll be out 7 years with no standard of care. She's what we in the stratosphere of survivors with this kind of tumor. Is she doing well? Reasonably well. She has a little depression. She's just got a wheelchair. She's very upset about it. She's not. We don't have the cure for this disease. What we've simply done is slowed it down so people can live longer and not be so debilitated. Then we started to do testing. We started to use her as our test. And she said, yes, I'll be your guinea pig. Please take the information from me. So we started to tell, okay, stop, go on the ketogenic. She did all this. You see her blood sugar is going down here in light blue, and the ketones are going up. And each one of these points is every day, 2 or 3 times a day for every month for a 2-year period. Her fingertips are all like, like a pincushion, but she got us all. Now, we started categorizing all this information. Then we found out that how are we going to translate this to make it easy for people in clinics and physicians to work with these people? It's going to be hard. She gets in, she has a handicapped parking spot, right? Some guy pulls in and takes her handicapped parking spot. She gets freaked out. All upset. She goes upstairs and measures her blood sugar. It's 165 milligrams per deciliter. She emails me, I'm dying. Here I'm back in Boston College, and she's from Nice, France, telling me. And I said, what's your ketones? Oh, they're still pretty high. So I said, oh, they are. So I said to my students, why don't we make a formula? Why don't we try to figure out, because these people I'm telling you right now, stress management is extremely important when you're dealing with cancer patients, because the disease itself causes stress. It's an emotional thing. There's something about having a fatality in our existence that knows that, hey, we don't have all the tomorrows that we think we're going to have. That creates stress. Stress elevates. Blood sugar goes up as the result of that. You've got to have stress management. You're going to do much better. So we decided to redefine the nature of this. Glucose ketone index calculator. Published the paper in Nutrition and Metabolism. Take the ratio of blood glucose in millimolar and ketone, and we get the ratio. And we think by meta-analysis in our own studies, 1.0 or below seems to keep these tumor cells in a lower rate of growth. So we published this. And we think this can be used by the cancer, not only brain cancer, all cancers, because they're all basically the same disease. They're all fermenting. They need glucose. And look at when we replotted her data. See how nice? Now the smooth curve. So even if she gets into a fight with the neighbor, it's not, the ketones are keeping that number down so she doesn't freak out. Now here, what's going on here? Ketogenic diets done correctly are powerful weight loss therapy. This is what people are using it for, basically. Right? They're all talking about ketogenic diet. You can lose weight. You don't eat any carbohydrates. The weight flies off you. Trudy, the person told me, he says, you can never be too rich or too thin. But she reached the point where she says, I need to put on a little weight. So she gets off the diet a little bit, and then she goes back on it, and she's doing this. Because she's thin, but she's healthy. She's thin but healthy. But anyway, it's a weight loss diet, so you have to know how to balance all these things out. The bottom line is that you got to lower blood sugar and elevate ketones to hold this beast down. It's not going to be as angry or growing as rapidly if you can deprive it of its fuels. Now, how are we going to manage the disease long term? How are we going to call it resolution? Okay, cancer resolution. What does that mean? Okay, that's a long-term management. Okay, we want to keep people alive longer and healthier than what is currently done. Is it possible to develop a therapy that will provide long-term resolution without toxicity? Okay, that's the key. That's the goal. That's our goal. Okay, now, we know that these tumor cells are filled with mutations. The genome projects tell us that. Most of the cells in the cancer have all these mutations. They lack the ability to flip from one form of energy to another form of energy because they have all these genetic defects. So if we begin to change the environment in which they live, their microenvironment and their own ability to grow you need energy. You need something to fuel. What fuels their growth? Glucose and glutamine. They can't grow without those molecules because they can't respire. They have to ferment. Their respiration is defective. So what we do is we deprive them of their fuels that they use to ferment, and at the same time we force feed them respiratory fuels in the presence of oxygen. And we try to blow them up without harming the other cells in the environment. All the other cells are burning ketones. So they're going over to this natural fuel that we evolved to use because they have good mitochondria and they're healthy. The tumor cell is now, they always say he's tough, he's fit, he's strong, he's resilient, because you never take away the fermentable fuels that cell needs to grow. The therapies that we give cancer patients make the fuels that cell increases in the environment. This is nuts. And the drugs that we use to kill cancer cells are targeting the nucleus. The nucleus is not the problem. Right? So what we do is we bring them into these restricted conditions called the press. You suppress the growth. This kills off many tumor cells, but not all of the tumor cells, because this targets glucose, but not glutamine. And I've seen raw restricted vegan diets work also well. But it doesn't cure cancer, it slows it down. Now, I have to admit, there are some people whose cancers respond remarkably well just to the press. I've seen it. But do you get long-term resolution for everybody's cancer simply by putting them on a ketogenic diet or a raw vegan diet? No. Because we don't control the other fuel, which is glutamine. Non-toxic drug targeting of glucose and glutamine and hyperbaric oxygen. And hyperbaric oxygen becomes important because radiation, which is used in many, many forms of managing cancer, kills cancer cells by increasing ROS, R-O-S. But it's indiscriminate killing. It kills the tumor cell, it kills the neighbor, it kills a lot of other things, and it creates necrosis and frees up glutamine. We don't want that. Hyperbaric oxygen can do the same thing without harming the normal cells. As long as you can deprive the tumor cell of its fermentable fuels, then it becomes susceptible to ROS, created by hyperbaric oxygen, which is much less harmful to the body than is radiation. Now, exercise is an important component, but we don't yet know how to balance the exercise issue in with these other processes. This is called the cocktail. The cocktail is going to involve the press and a series of pulses that are going to be delivered to people over a series of time, and we would like to make this time the same time frame that you would do if you had standard of care. So they give you a whole routine of radiation, they give you a whole bunch of chemo, you come back for this and that. We think we can build a cocktail that will have the same therapeutic benefits to resolve the disease, make people live longer, and quality of life go up, because you're not making these people sick. You're essentially making them healthy. The only thing you're making sick is the tumor cell. The normal cells are getting healthier and healthier. So this is the strategy. And we just have a preliminary results. This is using 2-deoxyglucose. It's a glucose inhibitor with the ketogenic diet. The drug has no effect in very low dose. The diet has an effect. Diet and drug, you get synergy. This proving that we can get synergy by using diet and drug. And then, of course, Dominic D'Agostino, my colleague, was here last time, and he presented this from Angela Pope. We all worked together on this. We label the tumor cells bioluminescent labeling so we can follow their metastasis through the whole body. Beautiful model, excellent model for systemic metastasis, which is the killer of most people. Hyperbaric oxygen by itself, well, not much. Ketogenic diet by itself, yes, a little bit, yeah, but this wasn't restricted. It wasn't restricted. If you restrict it, you get much better results. And then, of course, if you put the two together, you get synergy. And this is the way, this is press pulse. So in this particular case, it's the diet with hyperbaric oxygen. Also, you have to realize 25% of people that die from cancer get metastasis to the brain. Lung to brain, breast to brain, kidney to brain. So brain becomes a big problem. So this mouse has a very bright body, very purple. And of course, the brain it came from the viscera to the brain. This mouse has the same bright, not as bright, of course, but the brain was significantly reduced in cells that entered the brain. This is just the diet. We're working on new formulation. This is another thing. We have so much to do in this field. It's unbelievable. We can tweak these lipids and we can make these proteins different, every one of them designed to stress out these tumor cells while making the normal cells healthy. It's just a world of unbelievable new stuff that could come down if people were interested in it. Can't get these people out of this dogma. It's unbelievable. Now, this is a concept slide that only the biochemists would see. For a cancer cell to grow, you got to make protein, you got to make fat, and you got to make nucleic acids. You got to make that. Otherwise, you can't grow. You have to rebuild everything. Glucose and glutamine are the fuels to build the machinery, to build the nucleic acids, the fats, and the proteins. The cell is blown out, so it has to use these primitive forms of energy. So if you shut these down and give them hyperbaric oxygen, you're going to blow this sucker up. He's not going to be able to protect himself. These cancer cells have powerful antioxidants. They are very, but they're on the edge of life. They're this far from death, but they have a very powerful antioxidant system made from glucose and glutamine. If we take away that, their shield of protection evaporates, and we starve them, and we put the oxygen in there, we blow them up, and we make it so it's non-toxic to the rest of the body. Now, I just want to end with this one slide. Somebody asked me, what about the dogs? Okay, I think the segue into the human is going to come through the canine. Okay, so this woman contacted me from Georgia with her dog as a giant mast cell tumor right under the nose. She goes to the vet. The vet says, we're going to surgically resect the tumor. We're going to give the dog radiation and chemo. What else is new? The dog might live 6 months, going to be sick. You have to realize that. She said no. She said, I couldn't believe, she just looked at my YouTube videos and read the papers. A layperson that has no, she went to the butcher, she got organic chicken meat with the bone still in it, medium chain triglyceride oil, little fish oil, and a raw egg, and said, okay, this dog weighs this much, it's a working dog, it should have 1,800 calories a day or something. I'm going to cut it by 40% and give the dog 2 meals a day of a restricted amount of food. The dog lost 5 pounds in this treatment. But look at this. It's July 2013, September, this giant tumor went to that spot. Now here in 2014, it went here. In January 2015, the dog has just this tiny little freckle right there. The dog, I called her up just a couple of weeks ago. How's Minka doing? Oh, Minka's doing fine. So now the guys from Quest Nutrition saw this stuff, and they built this keto pet sanctuary in the middle of the Texas desert, and they put in vets and MRIs for dogs, hyperbaric chambers for dogs, ketogenic diets for dogs. And they presented the work at the meeting that we went to in Tampa, and they were able to resolve cancer in 12 out of 15 dogs, just using metabolic therapy. And they're going to publish it. They got a bunch of physicians and scientists that are working on this. And I says, can you imagine if this stuff gets out into the literature? Why are we curing all these damn dogs of cancer, and we're all being nuked and poisoned? Does that make sense? Anyway, to conclude, cancer is more of a mitochondrial metabolic disease than it is a nuclear disease. I hope I was able to convince you, or at least open your mind to that. Preclinical and case report studies indicate that the restricted diets can be effective non-toxic press therapy for managing malignant cancers in children and patients, adults. Effective pulse therapies can include oxygen, glucose, glutamine targeting, and a whole variety of other things that we have just begun to look at. The glucose ketone index is simply a monitor that allows patients to feel good about themselves and at least to say, I'm fighting this disease with my own capabilities. I'm not relying on the pharmaceutical industry to tell me how I'm supposed to fight my disease. Now, I've worked with a lot of physicians and scientists on this. from all over the place. I've been funded by private foundations, and I want to mention the Single Cause, Single Cure Foundation. And if you guys, I don't take money, if you want to give money to this, this is one of the few foundations that actually is interested in trying to manage cancer using metabolic therapies. They're not so interested in the mechanism, they want to see results. Is it possible to do what I just said? Build cocktails that are non-toxic, that kill tumor cells while making the rest of the body healthy. So they're interested in hyperbaric oxygen, non-toxic drug targeting, and see if we can, in fact, resolve these diseases, because we're going to move it right. I have a number of physicians that want to move this right into the patient population. If it works in these preclinical models, because we don't want to be treating these mice and having them die and suffer just like humans are dying and suffering. So if I can keep these mice alive with metastatic cancer and glioblastoma for a significant period of time, and they look pretty good, and they're not dying, then I'm going to tell my friends in the clinics, why don't you try this on your patients? So thank you very much for your attention.